Can you hear me in the back? Is that okay? If the mic is not working? Okay, so I'll set up with the mic. We're going to start here with this slide. So this is the last portion that we're going to cover today, the last content that we're going to be covering. And we're going to talk today about transmission, synaptic transmission. So last time we talked about the conduction of the action potential, right? So if we have time, I'm going to try to get back to that at the end. So to make sure that everyone understood that. Testing?
You got it yet?
Testing? So as you see here, there are three processes that we've been discussing. We talk about the action potential, which is generated when there's enough stimulation reaching threshold, right? When there's that amount of depolarization reaches threshold, that triggers the action potential, which happens here at the axon hillock. And remember, that segment of the membrane is where the sodium and potassium channels are expressed. That's why you can generate the action potential in the axonal membrane. Okay? So that's the process of generating the action potential. Now remember, there are inputs that are coming in the dendrites, and those synaptic inputs, if there's enough depolarization reaching here, the axon hillock, that will trigger the action potential. Okay, so you have multiple excitatory inputs in the dendrites, but also you have inhibitory inputs. So it depends. Now the computer. Okay, we're back. Okay, so it depends, again, on the amount of excitation and the amount of inhibition if there's enough depolarization to reach threshold, to get into that threshold. So that's what we call the postsynaptic potential. So postsynaptic potentials, they're going to be generated across the dendrites, and that depolarization or inhibition will get here to the axon hillock and enough. And again, if there's enough depolarization, then that will trigger the action potential. If you have more inhibitory inputs, that will reduce the amount of depolarization, that won't reach threshold, then no action potential will be triggered. Okay? Does that make sense? You guys with me?
Yes.
Okay. So we have conduction taking place here in the axon, and eventually, as you see here, we have the presynaptic terminal. So we're going to talk about what happens here at the presynaptic terminal. That's what we call synaptic transmission. And we'll also talk about synaptic integration. So that means integration of synaptic inputs, especially that are coming here. And those inputs, again, will summate together, will add up together. And if that's enough to reach threshold, then we get an action potential. Okay, so we have two types of synapses that are in the human body. So mainly, or we call them the electrical synapse and the chemical synapse. Okay, so the electrical synapse and the chemical synapse. Now, mainly we're going to talk about the chemical synapse, and we call the chemical synapse because it releases a chemical, a neurotransmitter, right? So specific neurotransmitters are going to be released in response to that action potential reaching the presynaptic terminal. One more time, the action potential is coming down the axon, and it's going to go into the dendrites. Into the presynaptic terminal. Once reaches the presynaptic terminal, that will promote the release of the neurotransmitter. And then the neurotransmitter is going to bind to postsynaptic receptors. Okay, so we have the presynaptic and the postsynaptic. The neurotransmitter is going to be released from the presynaptic due to the action potential reaching the terminal. Okay, so we talk about conduction of the action potential. Why is that? Because at the very end, it will reach those terminals, those presynaptic terminals, and that promotes neurotransmitter release that communicates with the other neuron. So we call this the synaptic cleft. Synaptic cleft, that's the space between two synapses. Okay, they're not joined together, they're separated by that space. That's the synaptic cleft. So we have presynaptic, vesicles, neurotransmitters, those are released by exocytosis and then you stimulate those postsynaptic receptors. That will trigger then a chemical change or an electrical change here, an electrochemical change, because you have the binding of the neurotransmitter, and then that will cause ions to flow in. You guys following? Okay. So the electrical synapse basically is because it's joined by gap junctions that allows the ions to flow through. So again, we're not going to talk about the electrical synapse in the central nervous system. We're going to mainly talk about the chemical synapse. Okay, so the chemical synapse, again, you have pre and postsynaptic. This is the presynaptic terminal, and you know it because there are vesicles, right? That they store the neurotransmitters. And here, this black portion here represents the postsynaptic density, which is basically the postsynaptic receptors. Those are in the membrane. Okay, postsynaptic density or postsynaptic receptors. We talk about the synaptic cleft, that's the space in between the pre and the postsynaptic membranes. And there are different types of neurotransmitters that we're going to be talking about. So, for example, a cell that produces dopamine will only release dopamine. We call that a dopaminergic neuron. A cell that produces norepinephrine, how do we call that one? Noradrenergic, right? A cell that releases GABA is called GABAergic, then glutamatergic, serotonergic, and so on. Okay, cholinergic, that will be for acetylcholine. Okay, so that means that that neuron produces that particular neurotransmitter, and again, in response to an action potential in the presynaptic terminal, that neurotransmitter is going to be released. Okay, so let's talk about the different steps in order to have that synaptic transmission. So that process, again, of communication between two synapses, that synaptic transmission. You guys have this diagram? Yeah?
On the last slide, what was the fill-in?
Action potential signal transmitted across synapse via what? You tell me.
Oh,
Yeah.
okay.
Neurotransmitter. Okay? Neurotransmitter.
Thank you.
Yep, no problem. Okay, so here we have the presynaptic terminal. And we're saying that the action potential is coming down the axon, right? It's coming through that membrane, the axon. So this is the axon, and the action potential is coming down into the presynaptic terminal. Now, in the presynaptic terminal, there are voltage-gated calcium channels. I know we haven't talked a lot about calcium yet, but in the presynaptic terminal, calcium is going to be very important. So calcium is going to play a role in the release of the neurotransmitters. So these are voltage-gated calcium channels, meaning that they open in response to what? Depolarization, right? We said that channels open in response to the membrane becoming depolarized. So that depolarization causes the channels to open, and once the channels open, calcium is going to go into the cell, down its concentration gradient. Calcium then is going to interact with these proteins in the membrane of the vesicle. Okay, calcium, again, is going to interact with proteins in the membrane of the vesicle, and that interaction will lead to the fusion of the vesicle in the presynaptic membrane. So the membrane, again, of the vesicle will fuse with the membrane that is located presynaptically. And then that will allow the neurotransmitter to be released into the synaptic cleft. Okay? You guys follow? Any questions about that process? Okay, step by step, we have the action potential getting into the presynaptic terminal. That causes depolarization that will activate voltage-gated calcium channels. Calcium is going to go into the cell. Calcium will interact with the proteins in the vesicle, leading then to the fusion of the vesicle with the presynaptic terminal, and then that releases the neurotransmitter by exocytosis. There's no ATP involved, right? There's no energy involved. It's just the fusion because of that interaction of calcium with the vesicular proteins. Then, as you see here, the neurotransmitter, let's say dopamine, for example, dopamine is going to be released, and then it will stimulate postsynaptic receptors. So this is the postsynaptic membrane. So dopamine will stimulate dopamine receptors that are in the postsynaptic membrane, and that will cause ions to go through. Okay, so ions are going to go through. Okay. So a little bit more about the presynaptic terminal. So here we have calcium. Remember, what activates those channels? Depolarization that is coming via the action potential, right? That depolarization is because the action potential is reaching the presynaptic terminal. Calcium opens, and then look at here, so we have some proteins like calcium activates or interacts with calmodulin. Calmodulin then is going to phosphorylate, so it's a protein kinase. It phosphorylates some regulatory proteins in the vesicle, and that allows for the docking, which is basically that the vesicle comes close or approaches the membrane, and then eventually it fuses with the membrane, and then the neurotransmitter release. So here it's important that you have calmodulin is activated by calcium, and then calmodulin is going to phosphorylate those vesicular proteins. So it happens in the presynaptic terminal. So this one I'm going to put it FYI, so don't worry about it. It just shows that calcium is going to promote that fusion. That's what matters. That's what I want to emphasize. There's going to be recycling of the vesicle, but we don't really care as much about that. Okay, now the neurotransmitter, we said that it's being released in the synaptic cleft, right? It will stimulate postsynaptic receptors. But there also has to be a mechanism to control neurotransmission. So you don't want that neurotransmitter to be continuously stimulating receptors. So there has to be a control. And a way to do it is by terminating the action of the neurotransmitter. And there are several ways. For example, let's take glutamate here. So glutamate, once released, it can go back into glia cells. Okay, this is a presynaptic, this is a postsynaptic. So glutamate can be taken back into glia cells, and it will be metabolized in the glia cells. Okay, so it can be recycled back or taken back into glia cells, and eventually gets back to the neurons. Another example here of terminating the action will be with the monoamines. For example, like dopamine, serotonin, they go back presynaptically. They go back into the presynaptic terminal. This is a reuptake mechanism. Okay, reuptake, meaning that it's going to be taken back into the presynaptic terminal. Do you know any drug that modulates serotonin or dopamine or norepinephrine to get it back into the presynaptic terminal? Any medications? Mm-hmm.
They're like, I think it's surgically, the SSRIs.
Mm-hmm. SSRIs, SNRIs, they basically block that transporter. So what happens if you block the transporter?
It can't get reabsorbed. So
Right,
it's
so-
more in the synaptic cleft to preserve like levels of it for transmitting whatever.
Right, so you're increasing the amount of synaptic monoamines. By blocking that transporter, you're increasing the amount of serotonin that is going to be able to stimulate the postsynaptic receptor. So you're preventing that termination of the action. Okay? Acetylcholine. So acetylcholine is actually going to be break down by an enzyme. How do we call that enzyme? The cholinesterase. It breaks down acetylcholine. So in this case, that will be another mechanism, breakdown, and then choline is going to be recycled back into the presynaptic to restore acetylcholine. GABA also can be taken into the glia cells or can be also taken back presynaptically. So again, there are different ways to terminate the action of the neurotransmitter. One will be enzymatic breakdown. That will be the acetylcholinesterase. That will be enzymatic breakdown. It could be, for example, take it back into glia cells. That's another mechanism. And then another mechanism will be take it back in those reuptake, by that reuptake mechanism. It's like a recycling mechanism, getting back presynaptically. Okay, so that will control the amount of neurotransmitter in the synapse so that there's no continuous stimulation. Mm-hmm. And going into the glia cells. So moving away from the synaptic cleft into the glia cells. So we have that once that neurotransmitter diffuses across the membrane, right, it's being released, it will stimulate postsynaptic receptors. Let's say dopamine, it could be glutamatergic receptors, it could be cholinergic receptors, so different type of receptors. And then that will allow the flow of ions. Okay, so once that neurotransmitter binds into the postsynaptic receptor, that postsynaptic receptor can open or change its conformation, and then ions are going to go through. That will be the case of ligand gated. That will be an example: GABA here, AMPA, and NMDA. These are glutamatergic receptors. So when glutamate binds to the receptor will open, and ions are going to go into the cell. The ions will depolarize the membrane. In the case of GABA, when GABA opens, which ion is going to go through GABA? So GABA is inhibitory, not glycine, but close. Which ion? When GABA opens, chloride goes through, and then chloride is going to do what to the membrane potential? It will make it more negative, because chloride is a negatively charged ion. So if you make the membrane more negative, will it be easier or more difficult to fire an action potential? More difficult, because you're moving away from threshold. That's why these cells that release GABA, they are inhibitory. Okay, so inhibitory inputs release GABA. GABA make the membrane potential more negative. So it moves it away from threshold. So more difficult to fire an action potential. Again, that's why GABA is an inhibitory neurotransmitter, because it inhibits the propagation or the generation of action potentials. So we have two types of receptors that we're going to be discussing. We have the ligand-gated ion channels. Okay, ligand-gated, that means that a neurotransmitter binds to the receptor, and then that causes the channel to open, and ions are going to flow. Those are ligand-gated. And then we have the G-protein couple receptors. The G-protein couple receptors. That means that when the neurotransmitter binds, there's going to be an internal conformational change that releases the G-protein, and that causes second messengers to be activated. Second messenger cascades of events. Okay? So we have two types, again, ligand-gated and G-protein couple receptors. So the ligand-gated ion channels, when they open, they allow ions to go through. The G-protein couple receptors, there's no ions flowing through. Okay? There's going to be an intracellular removal of subunits that are going to interact with other channels. But there are no ions flowing through. Okay? Here you have ions flowing through. With the G-protein couple receptors, there's no ions directly going to that particular receptor. Okay, so this is an example of both the ligand-gated. So following here, so we have a neurotransmitter binds to the channel. This is a ligand-gated, so the ligand, which in this case is this green neurotransmitter, it binds to the channel opens, and then ions, these ions are going to go into the cell. So ions flow across the membrane. If those ions, let's say they are sodium or calcium, what will happen to the membrane potential? Will depolarize, right? Because sodium, calcium, those are positively charged ions, so they will depolarize the membrane. What if we're talking about GABA? Let's say that this is GABA. If GABA binds to the receptor, which ion is going to flow through? Chloride. And chloride will take the membrane away from threshold. It will hyperpolarize the membrane. Okay, so that will be the inhibitory action. So that will be for ligand-gated. Now, G-protein couple receptors, so you have the neurotransmitter binds to the receptor. The receptor is going to release these subunits. You have the alpha, the beta, and the gamma subunits. Those are released. And then those subunits can interact with receptors that are nearby, or ion channels that are nearby. But there's no flow of ions through the G-protein couple receptor. Okay? It will lead to this cascade of events intracellularly that can activate nearby ion channels. So the neurotransmitter will cause either excitation, that would be glutamate, for example, or inhibition. That will be the case of GABA. And then when there's excitation in the postsynaptic cell, let's say that that neurotransmitter was glutamate, it binds to AMPA receptors that causes sodium to flow in. Sodium will depolarize the membrane. That's called excitatory postsynaptic potential. Okay, excitatory, again, because it's depolarized in the membrane. Postsynaptic, because we're talking about the postsynaptic cell. And then potential, excitatory postsynaptic potential. It's a depolarization of the postsynaptic membrane. Is that an action potential? An EPSP. Is an EPSP an action potential?
No.
No, it's depolarizing the membrane, but it's not an action potential. Remember, this is happening in the postsynaptic cell. There are no sodium channels or voltage-gated sodium channels or voltage-gated potassium channels there. So we're depolarizing the membrane, but we're not really triggering yet an action potential. So you have EPSPs, excitatory postsynaptic potentials, and depending on the amount of those, if they reach enough depolarization at the axon hillock, then the cell will fire. Okay? Clear? Okay, so we have depolarization of the membrane. That will be with the EPSPs. So excitatory postsynaptic potentials, and you can see here they are related, let's say glutamate, for example. So glutamate binds to the receptor, that allows a conformational change. Sodium is going to go into the postsynaptic cell, that depolarizes the membrane. So here is an EPSP. So there's a depolarization of the membrane. But remember, outside in the postsynaptic dendrites or in the postsynaptic neuron, still there are no, in that location, there are no voltage-gated channels. So you have depolarization, but it still is not able to trigger an action potential in the dendrites. Okay, so that depolarization is going to start to come down near the axon. So you have the EPSP, and the EPSP, the amplitude is going to be dependent upon the amount of receptors that are open. So the more receptors that open in response to glutamate, the more sodium that comes in, the more the depolarization. So in comparison to the action potential, remember the amplitude of the action potential? How do we call it, that characteristic about the action potential in regards to the amplitude?
It's the same.
It stays the same. There's a term that we use, all or none. Once you trigger, it's all or none. So they all look the same. The amplitude is the same. With excitatory postsynaptic potentials, the amplitude, again, is going to depend upon the amount of stimulation. The more the neurotransmitter, the more the receptors that are activated, the more the sodium that goes in, the more the depolarization that takes place. So this is what we call graded potential. It's a graded potential. Okay, it's graded because it depends on the amount of stimulation that is receiving. The action potential, on the other hand, is all or none. It's non-graded. It's always the same. So it doesn't depend on the stimulation. Now in the action potential, we said that if there's more stimulation, how is that encoded? Frequency, right? The more the stimulation, the higher the frequency. They fire very close together. Okay, so we have the EPSP. Remember, this is taking place in the postsynaptic cell. Once the neurotransmitter is being released, binds to the postsynaptic receptor, that causes an excitatory postsynaptic potential. Okay, I'm going to skip on this one. So this we covered already, right? We're saying that now for these potentials, the greater the frequency, there's greater entry of calcium, right? If you have more action potentials coming together, you have more calcium. That means that more vesicles are going to be fusing to the membrane, more neurotransmitter. Okay? So again, the neurotransmitter release is going to be dependent upon the amount of action potentials that are reaching the terminal. So the frequency of the action potentials will result in a greater stimulation of the postsynaptic cell. Okay, so greater frequency of action potentials here will end up leading to more stimulation of the postsynaptic neuron, because you have more neurotransmitter being released. Is that clear? The more the frequency of action potentials reaching here, the more of this mechanism that is being triggered, meaning the more vesicles that fuse, the more neurotransmitter release, then that leads to more stimulation of the postsynaptic cell. Okay, so we talk about graded potentials. So remember, the EPSP is graded. It depends on the amount of stimulation. The higher the stimulation, the greater those potentials are. And if we are here in the axon hillock, then it will trigger an action potential. Remember, the action potential will only be triggered at the axon hillock. But that amount of stimulation, again, will depend on the inputs that the cell is receiving. The more inputs, the higher the EPSP. Okay, EPSPs are conducted through the cytoplasm. So again, this is a postsynaptic cell here. So you see here the inputs are coming. So those synaptic inputs are going to get and travel, and then eventually they have to reach the axon hillock. The more depolarization, the easier to fire an action potential. But remember, you have also inhibitory inputs. So you have excitation and inhibition, and it depends on how much of that depolarization reaches here the axon hillock. If you have enough to reach threshold, then an action potential is produced. If you have more inhibition, that reduces the amount of depolarization, then that won't be firing an action potential. And remember, the action potential, once it's triggered, it will travel down the axon. So why the action potential cannot travel back into the soma? Why is that? What's that? It's only anterograde, but why?
There's no gated, voltage gated.
Right, but why here are, so why the action potentials when it's traveling down the axon, it cannot travel this way? You have here voltage gated channels. No, it's not due to the hyperpolarization.
Is it because of the myelin?
No. The sodium channels, remember, in the patch of membrane that just depolarized and is repolarizing, the sodium channels are inactivated. So there's the refractory period. So you cannot go back. Okay, that's why it cannot go back, because the sodium channels are inactivated. So there's a refractory period, and you cannot fire that patch of membrane. It will only travel down the axon. Okay, so again, you're getting all of those EPSPs, excitatory postsynaptic potentials, and if you get enough depolarization here, then the action potential will be triggered. And you have the concept of summation. Okay, summation. That means that those postsynaptic potentials, they will add together. They can add together to the depolarization to make it greater in order to reach threshold. Okay. So we have excitatory postsynaptic potentials, but also we have inhibitory postsynaptic potentials. Okay, you have both excitatory inputs and inhibitory inputs. Now, inhibitory inputs are coming because of GABA binding to the GABA receptor. That causes chloride to go into the cell, and chloride will hyperpolarize the membrane, will make it more negative. So take a look here. This is an IPSP, inhibitory postsynaptic potential. So you see that it's hyperpolarizing the membrane. So the EPSP, on the other hand, was depolarizing the membrane. This one is hyperpolarizing. So again, depending on the amount of inhibitory inputs, that will determine the amount of depolarization that finally gets into the axon hillock. So here you have an example. So here is an IPSP. So this is an inhibitory postsynaptic potential. It's causing inhibition, right? It's causing that movement of the voltage to make it more negative. So if you give an excitatory input, what will happen to that excitatory input? Won't be able to reach threshold, right? So the inhibitory input is preventing the excitation to reach threshold and fire an action potential. So here you have the same excitatory input. So the same excitatory input was enough to trigger an action potential. But if that same input that is excitatory is preceded by an inhibitory postsynaptic potential, it will move it more negative, So it's more difficult now to reach threshold. You guys follow? So the same excitatory input here was able to trigger an action potential. Here, it didn't. Why? Because it was preceded by inhibitory inputs that were making the cell more negative. So they were moved away from threshold. It was more difficult. In order to reach threshold, you will need to give stronger depolarization. Okay, you will have to do, for example, more excitation so that these can depolarize enough to fire. Okay, so let's review this portion, synaptic transmission. And again, when we talk about synaptic transmission, we're talking about the events that happens at the synapse, presynaptic and postsynaptic. So I want you to make sure that you understand all of these steps and that you can explain them. Okay, that would be a good short essay question. Right? So make a line, make a circle, make a star, whatever you want to make, but make sure to be able to answer that question. So if I ask you, explain step by step the processes that takes place in synaptic transmission, this is what I'm looking for. Did you make the star? I didn't see anyone typing. I told you already. Okay, so the steps, remember, there's going to be an action potential coming down the axon. Step one, the action potential. If you leave out the action potential, I would be taking points away. The action potential is coming into the synapse. That causes voltage-gated calcium channels to open. Right? Calcium is going to go into the cell. Calcium is going to stimulate calmodulin. Calmodulin phosphorylates the proteins that are in the vesicle. That promotes that vesicle to start to move towards the membrane. It will fuse with the presynaptic membrane. Neurotransmitter is going to be released by exocytosis, and it will bind to postsynaptic receptors. Okay? So that will cause a biochemical and electrical response in the postsynaptic cell. Okay? So that will change the postsynaptic cell. It will cause the opening of channels or the activation of G protein coupled receptors. And then eventually, there's going to be removal of neurotransmitters. So what are three processes related to the removal of neurotransmitters that we discussed? So enzymatic degradation, that will be with acetylcholinesterase breaking down acetylcholine. That's one process. Going into the glial cells, so moving away from the synapse into glial cells, and what else? What's that? Presynaptic reuptake. Okay? Presynaptic reuptake. That's via the transporters. So it's that getting back into the presynaptic cell. So those are three processes. Another good question, right? Like mention those three processes that are related to the termination of the action potential of the synaptic transmission. Then you got those. Okay?
What did you say calmodulin phosphorylates again?
Yeah, so the calcium binds to calmodulin, and then calmodulin is going to phosphorylate the proteins that are in the vesicle.
Okay.
That phosphorylation leads the vesicle to move towards the membrane, fuse, and then release. Okay, so from this diagram, or from this, I can take two essay questions. Explain step by step what happens during synaptic transmission, and mention the different steps or different processes related to the termination of synaptic transmission. Okay, so those two questions potentially.
I know you just explained it. Can you explain the calmodulin, like where it's inserted into this?
Calcium will bind to calmodulin. Calmodulin will phosphorylate the proteins in the vesicle. Okay, so we talk about synaptic integration, right? We're getting all of those inputs, excitatory and inhibitory inputs. So those inputs are going to integrate together, and again, the outcome, the result, if there's enough depolarization, the cell will fire. If there's more inhibition, the cell won't fire. It will be inhibited, right? So those inputs need to integrate. So you're getting all of those excitation and inhibition. So how do they integrate together? So remember we talked about the postsynaptic. I'm going to skip this. No time for this. Oh. Okay, so integration. So both EPSPs and IPSPs, they can summate together. They can add up together. Okay? Remember, these are not action potentials. These are postsynaptic potentials. They can add up together. They are graded potentials. And we have two processes, spatial summation and temporal summation. What is a spatial summation? So a spatial summation means that you have close in space several inputs, and they can sum, the result of that depolarization can add up together, can sum together. So take an example here. So you have one single input, right? You have only one input. This is promoting depolarization here, so it generates an EPSP. Okay, you guys following? So here, a spatial summation means that you have multiple inputs, but they are close in space. So they're close together. So you have three inputs, they're close together. So that depolarization that is taking place will be able to add together. So now this is the EPSP. So what do you notice? That now the amplitude is greater, right? You have a longer duration. It also, the amplitude is higher. Will this facilitate firing an action potential when reaching the axon helix?
Yes.
It will be easier, right? Because now they sum together and you're closer, you're at a more depolarized potential. So that will be a spatial summation. And then we have temporal summation. So temporal summation means that one input is firing at a very high rate. So you have high frequency of action potentials. A lot of action potentials coming together. That means that more neurotransmitter is going to be released. That means that more receptors are going to be activated. So the depolarization here now is going to be greater. So these are two mechanisms related to integration of inputs. Again, spatial summation, multiple inputs that they're close in space. Temporal summation, one input that is firing at a very high rate. So you have multiple neurotransmitters being released, or many neurotransmitters being released, many receptors activated, so more depolarization. And the same question, will it be easier to fire an action potential at the axon helix? Yes, because again, you are adding together those excitatory inputs. Those are excitatory postsynaptic potentials. The same will be true for inhibitory inputs. So if you have multiple inputs close in space that are inhibitory, that inhibition will be greater. The same with temporal. An inhibitory impulse that is releasing a lot of GABA, stimulating a lot of GABA receptors, that causes, again, inhibitory temporal summation. Then it will go downwards, right? It will make it more negative. So spatial and temporal summation, that's basically the definition that we covered. So they are important to determine the strength of depolarization, and it's related to the frequency of the action potentials. Okay, remember here we have multiple in spatial summation, different inputs, multiple inputs that they are close in space. Temporal summation, one input, but firing at a very high rate. Let's see what time we have. So I'm going to skip the break because I know we have 10 minutes break at the beginning. Sorry. Okay, let's move on. So we're saying that here we have the inputs, right? We're getting EPSPs, IPSPs. So if you have enough depolarization here, that triggers the action potential, and that will communicate with another neuron. So remember, this neuron is receiving inputs. This is going to release neurotransmitters onto another neuron. So that will be the postsynaptic cell. So you have the neurotransmitter release, receptor binding, ion channels being opened. Ions are going to flow through. The postsynaptic potential is going to change. Excitatory or inhibitory postsynaptic potentials are generated. That leads to excitation or inhibition in the postsynaptic cell, and it will determine that summation, right? The more summation, the more, well, if you have more summation of excitatory impulse, more likely to do what? For the cell to do what? To fire, right? The more excitation, the more summation of excitatory inputs, the cell is more likely to fire an action potential. The more summation of inhibitory inputs, the the less likely to fire an action potential. Okay? So that integration of inputs is important to determine if a cell is going to be active or not. So again, think about drugs that modulate inhibition, that potentiate GABA. Those are going to reduce the firing rate, so they can be used to treat certain conditions. Like we mentioned about seizures, we mentioned about anxiety, for example. So you're damping down the excitation, you're reducing excitation by inducing inhibition, by modulating the inhibitory pathways. Okay, so here you have these different excitatory inputs. And remember, the input, the EPSP, is going to be relative to the amount of excitatory neurotransmission. The more neurotransmitters, this input will be higher, right? This is going to become higher. And if you have enough, then you have the action potential at the axon hillock. So remember, this is taking place here out in the dendrites, in the cell body. These are the excitatory postsynaptic potentials. Then you have the action potential traveling down and repeating that process in the next neuron. Remember, they're interconnected. Okay, let's talk about neurotransmitters. Actually, I think we have time, so maybe if you guys want to take a 10-minute break. You want to take it? Yeah, I see the faces. Okay, let's take a 10-minute break. Let's come back at 1:30. Ashley, you good?
It just doesn't spike
Okay, so we're going to move into neurotransmission related now to the different chemicals, right? The different neurotransmitters. So we're still in the topic of synaptic transmission, but now we're going to talk about individual neurotransmitters. And I want you to know like the main characteristics of those, okay? Because when we move into the treatment for different neuropsych conditions, we're going to modulate neurotransmitters. So most of the drugs that we have, they modulate the receptors, or they modulate the neurotransmitters associated to those receptors, or they modulate the excitation. So we're going to briefly talk about the neurotransmitters, and I'm going to try to make emphasis in the things that are more known about those neurotransmitters, okay? So when you think about serotonin, for example, what do you think about? Serotonin. SSRIs, you talk about the treatment of what? Depression. So that mood, right? The low mood. So elevated mood is achieved. One of the neurotransmitters mainly related to that is going to be serotonin. So we know that serotonin modulates that elevated mood. So we're going to talk about dopamine, serotonin. We're going to talk also about acetylcholine. So specific characteristics of those that are well known, well described, okay? That doesn't mean that that's the only unique, specific thing that they do, okay? Serotonin does a lot of things in addition to that. Feeding, it could also modulate to a certain extent the wakefulness as well. So there are multiple effects that these neurotransmitters do have. They modulate pain, but we're going to talk about the main things, okay? So that you start thinking about potential treatment in patients that have low levels of serotonin or dopamine and so on. Okay, so again, drug selectivity, that has to do with the fact that we can identify receptors and neurotransmitters. So the better we know the different neurotransmitters and the better we know about the receptors, the more we can be specific in terms of the treatments. So 4 characteristics related to neurotransmitters, they need to be localized. So usually neurotransmitters are going to be localized pre or postsynaptic. Mainly presynaptic in the vesicles, right? Now there are some neurotransmitters that can travel back from the postsynaptic into the presynaptic terminal. So there could be also some presynaptic localization. But the vesicles are going to be located presynaptically, okay? Release, that means that there's going to be a stimuli, and that stimuli is mediated via calcium. Remember, we talk about voltage-gated calcium channels. So calcium is very important, it's essential for neurotransmitter release. And then it will bind to specific receptor. So in order for a chemical to be categorized as a neurotransmitter, they need to be localized pre or postsynaptic, they need to be released by a calcium-dependent process, and they need to bind to specific receptors. So those three characteristics are related to what the neurotransmitter is. So we're going to, again, talk about the main ones. So we have the amino acid transmitters, we have acetylcholine, the monoamines, and then we have neuropeptides, nucleosides, diffusible gases or diffuse gases. That will be, for example, nitric oxide, and lipid-derived signaling molecules. Those are related to cannabinoids, for example. Okay, so we're going to mainly talk about these. Neuropeptides, nucleosides, diffusible gases, as nitric oxide, those we're going to see later in the semester. Okay, so the amino acid transmitters, those are the most prevalent in the brain. Okay, and those are related to what? If we're talking about amino acid transmitters, which ones do you think are going to be the most common?
Glutamate.
Right, glutamate and GABA. So you got it there. Glutamate and GABA. Those are amino acids. So those are the most common. Again, glutamate, the most important excitatory neurotransmitter in the brain. Remember the hierarchical system? That excitation, that if you modulate it, you're messing up with the excitation overall of the brain. And then you have GABA, the gamma aminobutyric acid, that's inhibitory. So in the case of GABA, inhibitory causes the opening of which channels? Think about GABA, chloride. So chloride channels. So it's a GABA receptor, but the GABA receptor complex has a chloride channel. So when the channel opens, chloride is going to go through. Chloride is positive or negative?
Negative.
Negative. So once it goes into the cell, it will cause the membrane to become what? Hyperpolarized or more negative, right? It make it more negative. So that's why it's inhibiting the cell. It's preventing the cell from fighting. So it makes the membrane hyperpolarized. Okay, so briefly, so for glutamate, so glutamate here we have a glutamatergic cell. So this presynaptic terminal is going to synthesize and release glutamate. Once glutamate is released, glutamate will bind to postsynaptic receptors, or we said before that it can go back into glia cells. Right? Do you guys have this diagram?
Yeah.
Yes. Okay. So glutamate is going to diffuse. Again, it will bind to postsynaptic receptors to have an effect, or it can diffuse into the glia cells. And there's a transport here, excitatory amino acid transport. So this is a protein in the membrane that will be able to take glutamate. Glutamate inside of the glia cell is going to be break down into glutamine by the glutamine synthetase. Okay, so the glia cells, they have an enzyme that will convert glutamate into glutamine. Now glutamine is going to be then released, and eventually glutamine goes back into the presynaptic terminal, and there it will be synthesized again into glutamate by the glutaminase. So you have two enzymes to learn, glutamine synthetase and the glutaminase. So one converts glutamate into glutamine, the other one converts in the neuron, converts then glutamine into glutamate, back into glutamate. That's the glutaminase. Okay? So that's like a recycled process. So again, termination of the action. So you don't want to have that glutamate all the time stimulating the receptors. So it moves into glia cells, gets break down, gets released, take it back by the neuron resynthesizes back into glutamate. Again, stored in the vesicle to be released one more time once the action potential comes down. Okay? You guys follow? You guys got those two enzymes? So if I ask you, okay, explain the metabolism or the process related to glutamate metabolism. So those will be like glutamate is released. Once released, gets into the glia cell. There's an enzyme called glutamine synthetase, breaks down glutamate into glutamine. Glutamine goes into the presynaptic terminal. Glutamine gets converted into glutamate by the glutamine synthetase, or glutaminase, I'm sorry. Glutaminase. Then you got glutamate to be released again. Okay? So there are several receptors that are related to glutamate. Those are going to be ionotropic. So ionotropic or ligand gated, that means that there's going to be an ion channel associated to it. So when the channel opens or the receptor opens, that will allow the entry of ions. That's why it's called ionotropic. Then we have the metabotropic. These are the G protein coupled receptors. So you can put here metabotropic or G protein coupled receptors. And a good way to remember is that metabotropic, think about like a metabolism, because it's an internal second messenger event that is being triggered. Right? G protein coupled receptors, there's an intracellular cascade of events, metabolism that is triggered inside of the cell. Okay, metabotropic, metabolism. G protein coupled receptors is the same. Ionotropic, again, when they open, ions are going to go into the cell. So we have AMPA and NMDA. AMPA and NMDA, those two are ion channels. They are ionotropic. So when AMPA receptors open, sodium goes through. And if sodium goes into the cell, that will cause what to the membrane potential? Depolarize it or hyperpolarize it? Depolarize it, right? Make it more positive. NMDA receptors will allow then calcium, so it's also depolarizing. Don't worry about magnesium and glycine. I'm going to leave that to Dr. Cardona. Just know that NMDA receptors allow calcium. And calcium also depolarizes the membrane. Okay, and this is important for long-term potentiation, synaptic plasticity. Again, that's going to be Cardona. She's going to cover that, which is associated to learning and memory processes. Okay, and then we have the metabotropic glutamate receptors. Again, these are the they're called N-gluars. These are G protein coupled. Okay, these are G protein coupled receptors. Mm-hmm.
So AMPA is associated with the depolarization and allowing sodium in?
Yes. So these two, AMPA and NMDA, both are ionotropics. When glutamate binds to them, they open. AMPA will allow sodium to go in. NMDA will allow calcium to go in. Okay? So this is a very complex diagram. I don't want you to memorize it. Just to know that there's events taking place in the postsynaptic cell, and you have the different receptors like NMDA, AMPA, and again, don't worry about memorizing it, but you start to see like different agents like alcohol, PCP, angel dust. They modulate NMDA receptors. Okay, they interact with NMDA receptors. That's why, for example, PCP could cause hallucinations because it disrupts NMDA activity in the brain. So we have here also ampakine. This is another type of stimulator for AMPA receptors. But again, don't worry about the drugs. I'm not going to be asking about the drugs. Just know that the better we know about those receptors, the more we know about the different agents that can modulate them. Okay, let's move into GABA. So with GABA, don't worry about the synthesis. Just know that GABA is going to be taken back into glia cells. So very similar to what we saw with glutamate. So then GABA eventually gets back, and that's resynthesized from glutamate into GABA. So glycine is also inhibitory neurotransmitter and is found in the spinal cord. Remember we talked about the spinal cord. Okay, in the cerebellum there's also glycine can be present, and other subcortical regions. So the most important one would be the spinal cord. Okay, the spinal cord. So we have GABA A that mediates fast inhibitory transmission, and then GABA B. So GABA B is a G protein coupled receptor. Remember, G protein coupled receptors, they don't allow ions to go through. It's more of an intracellular mechanism that is triggered. So these have a slow inhibitory component. And we were talking about a while back about baclofen, which is a muscle relaxant. Baclofen actually modulates GABA B receptors, not GABA A. So this one is ionotropic. This one is a G protein coupled receptor. Okay? This is ionotropic. This is the one that opens. GABA A is the one that opens and allows chloride to go in. GABA B is a metabotropic receptor, so it allows for that G protein to be released and stimulate receptors nearby. Okay? So ionotropic and G protein coupled receptor. Mm-hmm.
Can you repeat one more time with the
Glycine mainly in the spinal cord. Yeah, it's also the cerebellum and subcortical, but mainly in the spinal cord.
So just know.
It's inhibitory, yeah, and it's inhibitory. Just
Okay,
know
just know it's inhibitory.
inhibitory in the spinal cord. Yeah. Okay, so that will be for those two. And again, if you look at different drugs, like for example, benzodiazepines, alcohol, barbiturates, all of those drugs, they potentiate GABA. They increase GABA conductance. So you can see here also baclofen working at the GABA B. Remember, the GABA B is a G protein coupled receptor. Just this G subunit, and it modulates ion channels. Here the GABA A, this one is the one that allows chloride. So that's a good way to remember and to visualize it. And again, multiple drugs that modulate those GABA A receptors. Okay, acetylcholine. So we talk about the different nuclei for acetylcholine. We're going to call the basal forebrain, right? And we have two types of receptors. We have nicotinic receptors. Those are related to muscle contraction, and muscarinic. Nicotinic, again, related to the muscle contraction, and that's muscarinic, and then nicotinic, right? Nicotinic, again, related to muscle contraction, somatic motor neurons. There's also in the ganglia, like presynaptic versus postsynaptic, and these are the different nuclei. Again, remember we call it the basal forebrain, so don't worry about the different names. Only know that they are in the basal forebrain, which is here. Okay, so like the base of the frontal cortex. That's why it's called the basal forebrain. And then very important, acetylcholine, those nuclei that you see there, those are the ones that start to degenerate initially during Alzheimer's. So a patient with Alzheimer's has destruction or degeneration of cholinergic neurons in the basal forebrain. So those are the main projection neurons that get destroyed in the initial stages of Alzheimer's. And obviously that's degeneration, so eventually the whole subcortical and cortical regions start also to degenerate, also the brainstem, so there's problem with swallowing and eventually breathing, swallowing, so all of that feeding gets affected because of the brainstem being degenerated. Again, cholinergic neurons, very important in Alzheimer's. So we learn about the cholinesterase, right? It's an enzyme that breaks down acetylcholine. So if you were to design a drug to treat Alzheimer's, and you know that it's related to acetylcholine, what type of drug would you try to do? An inhibitor for the cholinesterase, right? If you inhibit the enzyme that breaks down acetylcholine, then you will increase the amount of acetylcholine, right? If you inhibit the enzyme that breaks down acetylcholine, then you have more acetylcholine in the synapse. So that will help the patient at least in the initial stage. Now, as the disease continues, there's going to be more degeneration, more damage to the fibers, more destruction of the synapses, so the drug will lose its efficacy. So that will be only in those initial stages. Okay, acetylcholine. So we talk about the action, the cholinesterase. So here's the enzyme. It's an enzyme bound to the membrane. It breaks down acetylcholine into acetate and choline, and then choline is taken back presynaptically to again resynthesize it. So choline again is taken back to resynthesize it. So acetylcholine has multiple actions in the motor neurons, and we talk about contraction of the muscle. That's the end plate potential. Preganglionic fibers. All of the preganglionic fibers release acetylcholine. The postganglionic in the parasympathetic nervous system release acetylcholine. Motor neurons in the cortex, in the midbrain, there are also neurons that produce acetylcholine. And it has excitatory actions. Okay, so remember, muscle contraction, that's mediated by acetylcholine. Now, there are some inhibitory functions, especially in the heart. This is in the heart. So that's via muscarinic receptors, not nicotinic. So nicotinic will be related to muscle contraction. Muscarinic receptors in the heart, they cause inhibition. So they slow down the heart. So it has an inhibitory effect. And notice how it works. So here's acetylcholine binds to the muscarinic receptor, type 2. It causes the release of the beta and gamma subunits, and this will bind to potassium channels. So it will cause a stimulation of potassium channels. So if you stimulate potassium channels, what will happen to the membrane potential? You stimulate or you open potassium channels, what happens to the membrane potential?
Hyperpolarization.
Hyperpolarization, why?
Because you're,
Potassium's allowed to flow out.
Because potassium is moving out, right? So potassium moving out, that means that the membrane becomes less positive, because potassium is positively charged. So more potassium leaving, the membrane becomes more hyperpolarized. So it inhibits or it regulates the muscle contraction in the cardiac system or in the cardiac muscle. Okay? So that would be for via the muscarinic receptor.
so
Muscarinic?
is inhibitory.
At least in the heart. So in the brain it can have different functions, but in the heart, I'm talking specifically about the heart, it has inhibitory actions. Okay, so this is a cholinesterase. Remember, the cholinesterase breaks down acetylcholine, it's a membrane-bound enzyme. And then multiple drugs that modulate the cholinergic system. So these, for example, are related to blocking the cholinesterase. So these are drugs that can be used in the treatment of Alzheimer's, for example. Tacrine actually is out of the market, but we have newer drugs that substitute that one. Atropine, we have some venoms also that can modulate, like the black widow spider venom or toxin. So don't worry about learning that, okay? Just know that we have two types of receptors. Okay, let's back up. Two types of cholinergic receptors. Nicotinic and muscarinic. Nicotinic are ligand-gated ionotropic. Okay, nicotinic are ligand-gated ionotropic, meaning that when they open, ions are going to flow through. And muscarinic, those are G protein coupled receptor, the one that I presented in the heart, right? It releases the beta and the gamma subunit, then modulates other ion channels. Okay, one more time. Nicotinic, that's ionotropic, that's related to muscle contraction, and then muscarinic is a G protein coupled receptor. So catecholamines, then we have within the monoamines, dopamine, epinephrine, norepinephrine, and then we have serotonin and histamine. So this pathway are the sites of many drugs. Stimulants like cocaine, for example, or amphetamines, they increase dopamine and norepinephrine, right? So the catecholamines, they are derived from tyrosine, and then we have the other amines like serotonin from tryptophan. Don't worry about the synthesis, you don't need to know that. So dopamine related to motor, very important for motor. So we talk about the substantia nigra, that's related to the motor control. Dopamine again regulates motor, so that's why when dopaminergic neurons degenerate in the substantia nigra, that leads to a motor problem, right? Motor performance related disease. Which one is that? Parkinson's, right? Like you have that slow initiation of movement, you have certain symptoms associated with like tremor at rest, right? And then in the BTA, remember we talk about substantia nigra and the BTA. So the BTA projects to limbic structures like the nucleus accumbens, and that's related to addiction. That's related to goal-directed behavior. Now, overstimulation of dopaminergic pathways, that leads to addiction, to like drug-seeking behavior, like going after the drug even though you understand that there are negative consequences. So that drive is because you're stimulating dopamine in this pathway, in the BTA to the nucleus accumbens. So that, again, relates to addiction or pleasure in general, because obviously that system is built up related to pleasure. The problem is overstimulation of the system, right? Because then you get addicted to that particular pleasure. Dopamine receptors are metabotropic, meaning that there's no ions flowing through. All of them are metabotropic, so highlight that. And they tend to have inhibitory actions in the brain. They're breakdown by two enzymes, don't worry about that for now. So FYI. There's the substantia nigra and the VTA. Okay, so these are projections all over the cortex. And then different drugs that can modulate dopamine, like cocaine. It blocks the transporter for dopamine, so it increases dopamine transmission. That's why it's an addictive drug. Like cocaine, it's an addictive drug. Heroin, addictive because they increase dopaminergic transmission. What else? So we have drugs that stimulate directly the receptors. That can be also used to treat Parkinson's. Norepinephrine. So norepinephrine mainly located in the locus coeruleus. Remember, we talk about that loci or nuclei. Don't worry about the lateral tegmental area. The one that I want you to remember is the locus coeruleus. So these are also metabotropic receptors. Do you know any noradrenergic receptor? You will get that in PDA, but you should know that at least. Like alpha receptors, beta receptors, right? Oh. Those are adrenergic receptors, right? Alpha, beta. They are centrally, meaning in the brain, but they are also peripheral. You have also peripheral receptors for norepinephrine. So norepinephrine, mainly excitatory, so mainly an excitatory neurotransmitter, and they facilitate many functions. Like neurotransmitters, like norepinephrine, have broad actions. For example, they are related to attention and arousal. Attention, arousal. So drugs related to ADHD, for example, they modulate norepinephrine. They modulate this neurotransmitter to help with the focus of attention. So attention and arousal, that will be norepinephrine. Pleasure, goal-directed behavior, addiction, that will be dopamine. Mood, that will be serotonin. Like acetylcholine, memory. Right? We talk about Alzheimer's. Well, memory, and also we talk about the motor control, right? With the nicotinic receptors. So don't worry about termination of the action for norepinephrine. There are enzymes that break down norepinephrine. But it's okay, don't worry, I'm not going to be asking about that. Different drugs that modulate noradrenergic pathways. So we have the SNRIs, serotonin, norepinephrine reuptake inhibitors, the monoamine oxidase inhibitors, propranolol, clonidine, alpha and beta receptors to treat anxiety. We have drugs to treat depression, okay, processing, that's the alpha-1 antagonist. So again, those effects are peripheral or central in the brain. And again, don't worry about learning those drugs. This is just for you to get a sense of modulation that we have pharmacologically. And then last we have serotonin. Serotonin, again, those are going to be mainly G-protein coupled receptors except for one. There's an exception. Serotonin type 3 receptor, that's the only one that is ionotropic. Okay, the only one that allows ions to pass through is going to be serotonin type 3. All the other receptors are metabotropic within serotonin system, within the serotonergic pathway. So serotonin, remember, mood, elevates the mood. It also modulates anxiety. It relieves anxiety, right? It reduces anxiety. Pain, it modulates pain, especially at the level of the brainstem and spinal cord. It modulates other functions like sleep, appetite, temperature, aggression. Okay, but again, the most important ones that I want you to remember: mood, anxiety, pain. Okay, that would be for serotonin. So serotonin, remember the graphite nuclei, that's the nuclei where those cell bodies are located. And different drugs that modulate serotonin, serotonin reuptake inhibitors, selective serotonin reuptake inhibitors, those are the SSRIs or the SNRIs. We have drugs that modulate serotonin type 3, this is used for the GI. Triptans, this is used for migraine. Okay, antidepressants, obviously. And histamine, So histamine, we haven't talked about histamine a lot, but we mentioned that histamine, so when you stimulate histamine, that promotes what? Sleepiness or drowsiness? I mean, sleepiness or wakefulness. If you stimulate histamine, that would cause what? Drowsiness or wakefulness? Okay. Drowsiness? Okay. Wakefulness? Okay, why wakefulness?
Antihistamines make you drowsy.
Right, the antihistamine makes you drowsy. So if you block histamine, then that causes drowsiness. So histamine normally, physiologically, is involved in wakefulness. Okay, so it's involved in wakefulness. If you block histamine, that causes drowsiness. Does that make sense now? Okay, so antihistamines, again, they interfere with histaminergic transmission. That means then that you're blocking histamine drive. That causes a blockade in the wakefulness, leading to drowsiness. Okay, so it's a neurotransmitter that promotes wakefulness. So you have it here, wakefulness. Also, it's related to memory, feeding behavior, arousal. Those are metabotropic receptors. Okay, and antihistamines, they have sedative properties, right? Because they're blocking, again, that wakefulness. Some of the drugs like TCAs, these are antidepressants. They also have antihistaminergic activity. Okay, and this is just to know where is it located. Don't worry about the location or the name of the location. Is it 2 mamillary? But don't need to worry about that. Okay, so as an overall review, quickly here, we have until 12:10. So what I'm going to do is the last 30 minutes, answer any question that you might have, or try to review things alongside, because the exam is going to be 2 weeks from now. So when the exam comes, it's going to be like already far away, right? So Cardone is going to start Monday. Remember, you have class on Monday, free on Tuesday. So the whole content of Cardone is going to be in the exam with this content. Okay, so I'm going to try to go back and forth with some questions. So here, like overall, remember when we talk about glutamate, GABA, you need to know where they're related from or released from. In the case of glutamate, those are released by which neurons? How do we call the neurons that release glutamate? Don't tell me glutamatergic, I know. Glutamatergic. Inhibitory? Excitatory. And how do we call those long axon neurons that release glutamate? They are excitatory. How do we call them? Projection or relay neurons. They have long axons. They send information about sensory, right, coming from the periphery into the brain, or motor, going from the motor cortex down into the muscles to innervate it, okay? So those long axons. And then we talk about also GABA. So GABA is released by which type of neurons? The local circuit or interneurons, okay? Another term will be interneurons. They release GABA. GABA is inhibitory, and then it will modulate excitation. Remember, anything that modulates these pathways is going to have an impact in overall excitability. Any drug that modulates GABA or glutamate will have an impact in the excitability of the neurons, of the brain. So dopamine, we talk about motivation, reward, addiction, pleasure, euphoria, right? When you have overstimulation of dopaminergic pathways, that leads to euphoria. And that's related to the addictive process, right? The high. That sense of high, that leads to addictive behaviors. Compulsion, meaning, again, perseveration. You go after the drug even though you understand that there are negative consequences. So dopamine, important for motivation, goal-directed behavior, addiction, all of those. And dopamine is located in which nuclei? Which two nuclei? Substantia nigra, which is related to?
Motor.
But the condition? Parkinson's. And then the other region is the VTA, which is related, it could be related to pleasure, and then the addictive processes. Okay, norepinephrine. So how's the name? What is the name of the loci where it is produced? The locus coeruleus. It's important for cortical arousal. So that arousal, that wakefulness, that attention. So think about attention, cortical arousal, wakefulness. It's also related to memory. Again, mainly arousal and attention, focus. Serotonin, where's serotonin located? The raphe nuclei, and then it's related to pleasure, right? But mainly think about like the mood, relaxation, like anti-anxiety. It has an anti-anxiety effect. Motivation, drive, so mood, okay? And then histamine, we talk about memory, other processes, but mainly wakefulness. Okay, wakefulness, so if you block histamine, that causes drowsiness. Okay, so I want to go back for a second. Let me see, I have a couple of questions here we can just review. Okay, let's see.
We don't need to know all this in the post where the kids are, right?
Okay, sure, I don't know what's going on. It's taking forever. Okay, questions?
Would we, with the different neurotransmitters, would we have to know if they're ionotropic or?
Yeah. So ionotropic versus metabotropic. So in the case of glutamate, we have AMPA and we have NMDA. Those are ionotropics. Okay. So you need to know those. Ionotropic, that will be AMPA and NMDA. For GABA, GABA A is ionotropic, GABA B is metabotropic. All noradrenergic receptors are G-protein coupled receptors, all of them. So no ionotropic. For serotonin, most of them except for which one? Type 3. Okay, serotonin type 3, that's the only one that is ionotropic. Dopamine, all of them are G-protein coupled receptors. Histamine, G-protein coupled receptor. Okay, so I would say, like, remember, for the cholinergic pathways, you have two types, muscarinic and nicotinic. Which one is ionotropic? Nicotinic. Okay, muscarinic is a G-protein coupled receptor. Let me see if I can restart it.
I think we get this. Okay.
You guys having trouble with the internet here? It was working fine, it's just now. No. Okay, any questions? Getting back into the different processes. We talk about propagation of the action potential. Any questions about the propagation of the action potential? So if we have an axon, right, we have an axon, and we're injecting current, that means, or it could be a synaptic input, or we're artificially poking with an electrode and injecting current. So that depolarization in the axon will cause the membrane to become what? Depolarized. That will cause opening of voltage-gated sodium channels. That will trigger in that initial segment, that will trigger an action potential. So how the action potential travels down the axon? Remember we said that this patch of the membrane is positively charged, so that will cause a passive diffusion of the charges inside of the axon that will then depolarize the segment up front. That depolarization causes the channels to open. That will trigger an action potential now up front. That will cause positivity. That positivity will move passively inside of the axon, will cause activation of voltage-gated sodium channels up front, and the process will continue. That's how the action potential moves across the axon. Remember, you're stimulating in one point, but that action potential travels down. Why? Because of that passive current that is flowing from positive to negative. That flow of current causes the voltage-gated sodium channels to open and continue the action potential down. Okay? The video that I presented to you, it shows very nicely the way it propagates. And remember, if you have myelin, then you don't need to depolarize that patch of the membrane. It will go faster. So that the distance that passive current will travel will go faster. That's why the myelin increases the conduction velocity. Okay? The portion about synaptic transmission. Any questions about that? Or synaptic integration? Remember, synaptic integration of synaptic inputs. We're adding inputs together. Summation, we have temporal and spatial. Who can explain temporal summation? Temporal.
It's like kind of the frequency at which it fires, and it makes the action potential larger.
So one input, temporal is one input, but it's very close in time. So the firing is very fast, as she said. The frequency is very high. That means that you have more neurotransmitter that stimulate that postsynaptic cell. So that allows them to add together. That's temporal. Very good. Spatial summation.
Multiple inputs at once.
Multiple inputs close in space, right? They're close enough so that depolarization can add up together. Remember, those, we call it graded potentials. Is the action potential graded? No. The action potential is all or none. Always the same amplitude. What codes or encodes the intensity of the stimulus with the action potential will be the frequency. Right? The more the stimulation, the higher the frequency of the action potentials. But always the same amplitude. With the postsynaptic potentials, those are graded potentials. So the more stimulation, the higher they are. The smaller the stimulation, the small those are. Okay? And that will be the case for inhibitory postsynaptic potentials. So which neurotransmitter causes excitatory postsynaptic potentials?
Glutamate.
Glutamate. Which neurotransmitter stimulates inhibitory postsynaptic potentials or causes?
GABA.
GABA. Which one? Oh, glycine in the spinal cord. Correct. Yeah. And then we talk about the role of calcium. I thought I heard calcium. So, what is the role of calcium in this whole synaptic transmission process?
It binds the calamine.
Can you?
Second messenger cascade.
Related to calmodulin. Yeah.
Phosphorylates the proteins inside the vesicle.
Right. So the vesicular proteins are phosphorylated. It fuses with the membrane, and then allows the release of the neurotransmitter. So that calcium-dependent process is essential for neurotransmitter release. Okay, so if you block calcium channels, that will prevent neurotransmitter release. Some of the drugs that we use in practice for the treatment of epilepsy or seizures are related to modulating neurotransmission with calcium channels. Like they block calcium channels, so you prevent neurotransmitter release. So you reduce excitation. Does that make sense? Like, as a mechanism? Any other questions?
Remember the confirmations for the voltage?
Yeah, so the different conformation of the voltage-gated sodium channel. So at rest, the activation gate and inactivation. Who can tell me? At rest, at the resting memory potential. Activation, open or close. Inactivation, open or close.
Activation close and activation open.
Activation, close, Inactivation open, correct. So at rest, the activation is closed, the inactivation remains open. As we depolarize the interior of the cell, sodium channels are going to start to activate, so that means that the activation gate is going to start to open because it opens with depolarization, remember? And there's a sensor in the receptor protein that will be moved because of the changes in positivity. So that will allow then for sodium to continue to go in. So during the upstroke of the action potential, what is the configuration of the voltage-gated sodium channel?
Open.
Again?
The activation.
Uh-huh.
Isn't
Sure.
both open?
We're saying during the upstroke of the action potential.
Are they both open?
No, they're both open.
Both open, right? Both have to be open so that sodium can go through. So that will continue to happen. They are open. And then at the peak of the action potential, what is the configuration?
The inhibition's closed.
What's that?
Inhibition closes.
The inhibitory gate or the inactivation gate closes, but the activation, is it open or closed?
Open.
Open, it's still open. So the inactivation closes at the peak, but still the activation is open. Can sodium go through?
No.
No, because the inactivation is blocking the channel, so it cannot go through. As the membrane repolarizes, what happens to the configuration? The activation starts to close slowly, and the inactivation starts to open. So eventually, when you get back to rest, activation is closed, inactivation is open. Okay? So those are the steps that I want you to know. Rest, upstroke, the peak, and the repolarization. Any other questions? Or any other concept that needs to be reviewed? Oh. I was anymore.
I'm creating a little spreadsheet.
Okay, let's take a look at these
Each
questions.
one paper.
You have the code,
Sketchy.
so you can participate. So there's the code. Okay. That one is tough. I like, okay, let's see. Okay, refractory period. Okay, you got it? I think I got it. This is AI generated, so sometimes like too tricky. Yeah, those are. I mean, obviously we tweak it and we change it, but this one I think I use it as it generated. Okay, let's go back one by one. So we're talking about refractory period. We talk about two processes with refractory period, right? We talk about absolute refractory period and relative. You guys remember? Sorry, what is absolute? You cannot, what?
Are officially inactivated, so you can't do another action potential.
Right, so you cannot generate a second action potential. And why? Because sodium channels are inactivated, or also you have potassium channels as well. So that's absolute. And then relative,
The sodium channels are reactivated, but you still have the repolarization of the potassium.
Mhm.
So if you have a stimulus that's high enough to bring it back up past threshold, then you can fire another action
correct,
potential.
correct, very good. Yeah. So those are absolute and refractory. Remember relative. Remember relative, as you said, if you have enough strong depolarization, you can get a second action potential. With absolute, you cannot, okay? So, let's see which one is the right answer here. I think it's B. You also thought that was B, so that's good. Yeah, so the slide title shutting down those potassium channels, and then the persistence of the inactivation for sodium. Very good. Let me see if I have anything else. Okay, let me see this one. Okay. So looking at this graph, which property of the action potential were illustrated here? Don't take pictures. Don't take pictures. Which property of the action potential are we illustrating? Which one do you think?
The all or none.
All or none.
Okay, which one?
All or none.
The all or none, right? We're not talking about refractory because we see that the action potential is actually propagating down, so there's nothing to do with refractory. We're looking at the characteristic of the amplitude. So you see that as the axon is depolarized via the action potential going down, moving down, you can see that it's moving from T1, and point A, point B, and point C, so it's moving, but it's maintaining the same what? The same amplitude, right? The same amplitude, so that's the all or none characteristic. Remember, if we're talking about PSPs, they're gonna start maybe here, so at least I can draw. So a PSP will start here, but as it continues to point B, and eventually it will decay. So as a postsynaptic potential, a PSP, inhibitory or excitatory, moves across the membrane, not the axon, but the membrane, the cytoplasm or the dendritic membrane, it will start to decay because the current is going to flow out. So that's what we call graded, because it depends on the stimulation, but also as they travel across the membrane, they will start to decay. The action potential won't decay. The action potential will always remain the same as long as it travels down the axon, okay, and reaching the terminal. So again, that will be the all or none characteristic. Very good. Let me see what else. Okay, let's do this one. We're looking at the permeability and the consequence of that permeability towards the membrane potential. Okay? And chloride should be negative. Sorry, I don't know what says positive there. Should be negative. Yeah, and potassium should be positive. It mixed it up. So I'm not trying to confuse you. I'm sorry about that. Yeah, I'm sorry about that. So think about potassium, positive, chloride, negative. You just mixed up the signs. That won't happen for the exam. You guys ready? So which one will it be? And remember, potassium positive, chloride negative. Mm-hmm. What happens if you increase sodium conductance? Depolarize the membrane. What happens if you increase potassium conductance? Mm-hmm. It's hyperpolarized, so the answer is B. Why does it hyperpolarize the membrane? Yeah, don't confuse it with the equilibrium potential. So why the membrane potential becomes hyperpolarized? It's related to equilibrium potential, but
Sorry.
more easy
Membrane
way to see it.
is essentially negative 9 millivolts. So
Say that again? Say it again?
potassium's equilibrium, the membrane potential essentially for calcium, or not calcium, for potassium specifically is negative 9 millivolts. So
That's equilibrium potential. Yeah, but think like simpler than that. A simpler explanation. Mhm.
It's the potassium leave the channels, the potassium leaving through the channels themselves.
So potassium is
So
leaving.
the cells becoming more negative.
Right.
the positive.
Right, so it will take, so if you relate your response to Catherine's, so it relates to eventually getting to that equilibrium potential for potassium, right? But the reason is because the channels are open, so potassium moves out of the cell, and since potassium is positive, you're leaving behind more negative. That's why then it becomes hyperpolarized, okay? So if you decrease chloride, what will happen if you decrease chloride conductance? That will depolarize or hyperpolarize the membrane. It will depolarize because you are closing chloride channels, so chloride cannot go in. If chloride goes in, that will hyperpolarize the membrane. So if I make this statement, C, increase chloride conductance, then that will hyperpolarize the membrane. Okay? And what about calcium? Decreasing calcium? Technically it can hyperpolarize it. But we're not going to really go into calcium. Okay, let me see how you guys did. Okay, very good. So most of you got it right. Let me see what else. Okay, any additional questions? I don't want to keep you hold you longer, but make sure to go over the content. I'm gonna be fair with the questions, so it's gonna be similar to what we've seen. I've been trying to repeat back and forward to try to integrate it. So if you have questions, let me know. You can email me. Also, you can stop by my office. I don't mind like going over the stuff. So the exam is gonna be 2 weeks from now, roughly. So make sure to ask any questions before that, okay? Thank you guys. I'll see you next year.